Good morning, everybody. Thank you very much for joining us today. The Hawaii Coffee Association is happy to host a series of webinars covering the latest research data concerning the coffee leaf rust disease in Hawaii. We are grateful to all the scientists whose work is helping our industry to battle rust and other diseases. Controlling coffee leaf rust from lab to the field is the topic of our webinar today, and it's going to be presented by Dr. Lisa Keith. She's a research plant pathologist at the USDA Agricultural Research Center in Hilo, with over 20 years of experience studying the biology, detection, and management of pathogens attacking coffee and other crops. This webinar will be recorded and published on our website. If you have any questions during the session for Lisa, feel free to leave them under the Q&A tab, and she will address them at the end of the presentation. Lisa will take it from here.
Okay, thank you so much, Shala, and thank you HCA for the invitation, and hello, aloha everyone online. So Ala did a great job with my title, Controlling Coffee Leaf Rust from the Lab to the Field. And before I begin, actually, I will give an incredible shout out to the amazing teamwork we have working with coffee. All agencies, the university, this work would not be possible without the support of the growers and the producers. So we thank you so much for that. I have an amazing team in the laboratory. A great shout out, especially with Blaine helping lead a lot of these coffee efforts. And just the laboratories, the colleagues, the friends we have at PBARC, at the university, the IR4 program, SHAP, and many others. And so this is the overview for my talk today. As a plant pathologist, I figure I always get the chance just to briefly describe how disease caused and what is so important about the pathogen, the host, and the environment. I figure today we're going to kind of give, I guess, an overview, some fundamentals, definitely some research data, but a lot of summaries. So I do have a lot of slides. It'll look like a lot of data, but I'll try to summarize for the best way I can, and then afterwards open it up for questions and discussion. But the second session, kind of go over CLR fungal facts. And then when we control CLR, what does it mean? What do these products mean? How do they work? And we'll give examples of what we do and how we do it, starting in the laboratory, working with potted plants in the greenhouse, moving to the field, and give you some summary and overall conclusions today. Okay, so this is one of my favorite slides, and you'll say, Lisa, yes, we always see this slide. But this is very important when it comes to how is disease caused in plants. And it is called the disease triangle. It has to have these three components working at the right time and the right place for disease to occur. So not only having the pathogen present and a susceptible host, but having a favorable environment. And this, as I mention all the time, it's not just a simple, well, is it the temperature? No, I mean, there's all encompassing environmental conditions. And so if something's too dry, even though you have the pathogen and the coffee host, you will not see CLR. And things we tend to talk about a lot are symptoms and signs of the disease. And just the reminder that the symptoms are actually how the plant, the tree, responds to pathogen attack. And the signs are actually what you're looking at the pathogen itself. Okay, so here's another slide that you have seen often. This is actually the disease, coffee leaf rust, the pathogen Himalaya vistatrix. And these are kind of they're not fun facts, but facts about this system and why it is so important. So the right side does show the disease cycle. The pathogen has spores that enter plants. They kind of really anchor themselves in. The fungus grows within the leaf tissue. It grows back out onto the underside of the leaf, produces spores, and the cycle continues and continues and continues. So some of the important words: obligate. Well, what does that mean? Well, this pathogen needs a living host to survive, which does make it challenging to study in the laboratory because you can't culture it on a plate. Okay, coffee is its only host, so it needs a living coffee tree, leaf, to survive. Stomates, why are they important? Well, this particular rust fungus enters only through the stomates, which are on the underside of coffee leaves. Okay, so spores may land on the top, but they have to work themselves down to the underside and enter through a stomate. It will enter through a stomate, it will grow within the leaf tissue, and it will actually grow back out through the stomate and produce the spores on the underside of the leaf. So these are just some examples of what the spores look like. They're grippy surface, kind of like Velcro. This right picture here is the underside, showing that powdery yellow substance that are the spores. So these are signs of the pathogen, signs of coffee leaf rust. Water is very important. It needs enough water to germinate. Rain splash is the typical way this pathogen spreads locally. So from the leaves to other branches to close trees, this is probably the main way. Physical activity like people, like animals can spread because these spores love to hitch rides and move. Wind does play a role, but not probably as big as other rusts. Spores have within infection, so a new infection starts, these spores will start to be produced by 2 weeks. So you can see how a very quick buildup can occur. And the spots or the lesions that you see on the leaves will actually continue to increase. So the center will become inhospitable for growth, and the fungus will continue to kind of grow. The spots will increase in diameter, and new spores will continually be produced from these areas. Okay, so to move into the goal is controlling coffee leaf rust. One way is through fungicide use. And this picture helps give the examples of various fungicides and how they work. And so just to go over this picture, you see the upper surface of the leaf, the lower surface of the leaf, okay, the leaf tip towards the right, the trunk of the tree to the left. You have the xylem and the phloem within the leaf and within the tree, okay? And here's the various, so the contact or the protectants in red, the droplet hits, it hardly spreads, it will control what lands on it, okay? Locally systemic is in blue. So when the droplet of the fungicide hits the leaf, it will spread a little. It will just start to spread within that leaf surface, and that's it. Okay, so again, what kind of protection are these products offering? Translaminars, when the spray hits the leaf surface, it will spread a little, but it'll actually move from, say, the upper surface to the lower surface, okay? But it is very limited. So we don't, I guess the true definition, we don't call it a systemic. It is translaminar from one side of the leaf to the other. Now, in black, it's showing a true systemic. So when it lands, it does spread a little, but what it does not only goes from surface to surface of the leaf, but will actually get in and move throughout the leaf and the tree. Okay, that's what's very important. Will move throughout. So the information on this slide to kind of compare the contacts, which is what our biologicals, the coppers are, to systemics. which also mostly includes then this translaminar, which is preaxor. Okay, the contacts are protectants. They need to be there to prevent infection. Systemics will actually cure if the pathogen is there. The contact essentially just adsorbs to the leaf surface while a systemic is absorbed, okay, and actually moves throughout the xylem and the phloem, where the contact is immobile. Where the droplet lands, it might slightly spread, but that's it. It's not going anywhere else, okay? Contacts all tend to have a low residual, which is why higher doses and more applications are needed, which is the opposite with systemics. They tend to have medium to high residues, so you can apply less and at lower doses. Now, contacts are good because they have multi-site modes of action, okay, which there are often very few problems with resistance, okay? Systemics tend to have a single site of action. So what part of the pathogen is this product going to poison, and how does it work? That is that mode of action. And with a single mode of action, there's a greater chance of resistance. And oftentimes you're going to hear, well, FRAC groups. Well, what are FRAC groups and why are they important? Okay, Fungicide Resistance Action Committee, companies got together, actually defined groups based on their mode of action. So that 7, the 11, products can have different names, but if the mode of action is the same, the FRAC group is the same. And that's, again, why you need rotation to cut down on potential resistance. Okay, so here's another way of looking at it, just to understand what to do, how to do it, and why it's important to work with the product the best way possible. Okay, so working from left to right, it's just the example. When you spray the fungicide and you have these droplets land, again, the contacts will essentially land and protect whatever area it is. Now, it really needs to be there prior to infection. The translaminar, you can see now with the fungicide, whatever leaves were received the application of the product, it will protect the leaf, okay? So hits leaves, it will protect that leaf. But what you can see in this last picture with the systemic, it not only goes up and down the plant, okay, in the vascular system, but because of that will actually offer protection to new leaves that are produced, okay? So this is what this blue is, how much of the fungicide gets on and in and continues to move. So that's critical to know. Will new leaves be protected? How do those droplets spread and move? And how much protection does it offer? Okay, so to get into fungicide testing, what we do and how we do it, again, from the lab. This is a picture of actual Himalaya spores, infected leaves, collecting spores for tests, and getting out into the field to see, does it equate to what we see in the laboratory? Okay, for laboratory testing, it is the best way, maybe the quickest way, to help prioritize what to look at in the field. Essentially, we collect fresh spores, we mix them in a tube with whatever product we're interested in testing, and we want to see, well, first and foremost, will it kill spores? Not every product works in that manner, and that's important to note when you're doing your testing. But with this test, you mix the product, the spores, you let them have contact for a certain period of time. We can plate them, incubate them in the dark because that's where germination occurs. Actually, when it is dark, for a certain period of time, it needs the dark. And then we stop the test and actually look at the number of germinated spores. And that's what this picture is showing. Some you see no germ tube formation. Here you see a lot of germination occurring. And this is how the spore that lands, especially on the underside of the leaf, will now this germ tube moves to the stomate and now infects. And you might have said, well, Lisa, you said this is an obligate. You can't culture it on a plate. For a very short period of time, we can get these spores to germinate on water agar to do these types of tests. Okay, so the first and foremost, it was really important to see what type of sanitation is best. And this is just to show you some of the initial data with the comparison of sterile distilled water and then the ethanols and the IPA and the bleach, when it came to shoes and tools and anything else moving through the fields, what you could use. And the time, within a minute, all of these products were very effective at stopping germination of these spores. Now, kind of an interesting note, this is the control, so it's just water. The spores are put in water, plated, and counted, and you can see that when they're in the water for a minute, there's a certain germination rate. When they're in for 24 hours, noticeably the germination rate decreases. So water's good for germination. A lot of water actually starts to be a bit inhibitory to the pathogen. Okay. Yes, this is a crazy slide with tons of data. This is really just to show you the constant action in the laboratory, testing any and all things we can get our hands on, whether that be the products registered for use, the coppers, the biologicals, Preaxor, collaborating with IR4 and testing the systemics to see what type of action they have on spore germination through these tests. Natural products, essential oil products, the sticker spreaders that we can use. And so I tried to summarize basically by these colors indicating what things are. So for example, Preaxor in this pink purple, you can see when you're looking at spore germination on this y-axis versus all of these different products. You can see Preaxor very effective at inhibiting spore germination, as are a lot of other things. Some of the systemics that IR4 is testing, excellent, but you can see certain others are not specific for this pathogen, which is good to know. Some of these others with the copper products, whether that be the one Cueva with detergent included or not, like Badge, quite effective. When you start to look at some of these other biologicals where double nickel, Serenade working well. We try to find some of these other friendlier products, maybe essential oil base, more for organic. It's a mixed bag, what it does to the spores, killing or not killing. And then you can actually see that some of these other things compared to this control, water control that we always run with the test, you definitely see variability. But keep in mind, some of the products don't work directly on the pathogen. There are products that stimulate plant defense. So this is not the most accurate way to look at those type of products, but we wanted to see maybe there'd be an added bonus of affecting spores directly. Okay, some other experiments. Now, sadly, we have coffee berry borer as well. So to get just a better understanding for compatibility, if you're mixing products, the Botanigard for CBB control and the various fungicides, same type of testing. You can make solutions following the label rates, mix them with fresh spores of Beauveria, and see how the products now, are they compatible or not? What happens? Do we see germination of the Beauveria spores? And it's a very similar, this is what you're looking at, water agar, and you can see the germ tubes formation, Beauveria spores germinating. Okay, so again, here's some graphs. There'll be 4 in a row, trying to summarize and to look more at the trends of the colors. Okay, so down here, it's the product combinations. Okay, and we're looking at the average germination of Beauveria. Again, we always have a standard water control and the various products, which I've grouped based on color. Okay, so the rate combinations, what you're looking at with these graphs are the product and Botanigard. If there's more than one rate on the label, it would be the low and the high rate of the product, and then the low and the high rate of Botanigard. So those 4 combinations. If the colors that you see, for instance, here with the control, there's only 2 bars, well, it's either the water control or the product only has 1 rate on its label. Okay, so right away you can see badge, whether that be SC, X2, mixed with Botanigard, germination, the Beauveria spores germinate well, as opposed to oxidate. Essentially, the combination will knock out the viability of Beauveria. Okay, just to go through quickly, Cueva, now another copper product with detergent, as you can see, definitely inhibits spore germination, and especially depending on the rate of the product. With the high Cueva rates, you see what happens. Essentially, it's knocking out the viability of the Bassiana spores. Okay, you have Problad, you have Cinerate. So here's an example of an essential oil product and what happens. Okay, the combination and the rates matter. Here's Capodac, a slight decrease. Okay, moving into, again, the IR4 systemic products, just to see what would happen. Well, you can see there's variability again. Now, these products are fungicides, so it does make sense that some of them will actually be inhibitory against the fungal product that you're hoping will control coffee berry borer. Excalia depends on the rate combination. Okay, here's a few more of the IR4 systemic products: Approach Prima, Appruvia Top, and then it depends here on the rate combinations of this last one. Okay, so that's the summary of some of the lab work that we do and how we do it. We're moving on. So a little more challenging to work with the living plant. But what is the next step oftentimes is to seeing what these products can do, how long they can protect plants using potted seedlings. And so here's just an example of some data for the treatments, looking at Serenade and Badge compared to a water control. And what we would do is take healthy seedlings and apply the treatment, let the treatment dry, and then inoculate fresh spores onto these plants the same day the treatment occurred, 1 week and 2 week post. So how long do these products actually protect the surface of the leaves? Take the average rating, looking at symptom development, looking for signs, those spores of the pathogen, once a week. And here's some of the results. So the controls, you can see within 4 weeks, definitely getting spots, visible spore production. And when you look now at this next slide, whether that be Serenade and Badge, or Badge, sorry, 4 weeks again, post-inoculation, symptom development and at variable levels. And you probably can guess based on the color of the residue, which product is rich. Okay, yes, more graphs. Trying to make it easier though, again, showing if we inoculate immediately after the treatment, 1 week, after the treatment was applied or 2 weeks after the treatment was applied. This is the week post-inoculation, that symptom development over time, and using a rating scale based on the number of lesions or the overall percentage of the leaf that's diseased. You can see the trends where I said, as far as with the water, already by week 3, you're starting to see those spots. 4, you start to see definitely, I mean, well, probably even sooner than that, within 2 to 3 weeks, spore development. And then the comparison of B for badge, S for serenade. There's a negative control, so making sure our plants just with water look healthy, and how long the protection lasts. And really, it's just the verification of what label states. You know, these products within, say, 2 weeks, 10 days, 14 days, and that's really what these trends are showing. I'm going to point out, let's see, this badge. Well, this was an interesting, wow, we sprayed badge, then we inoculated, and what kind of happened? You know, these other ones showing definite delays in symptom development, offering quite good protection up to that 2-week point. But right away on some plants, it was following more of a water control. Okay, this is why we can't stress enough why good spray coverage is key, especially when it's not a true systemic. Even the translaminar, where this product gets to, offers the protection for that spot, for that area. Okay, so the upper side of the leaf, you see spots, and the lower side of the leaf is where you see all of this spore production. When you spray, and in this case it was copper, if you spray the upper surface, essentially it's not getting to where the spores are. Okay, the lower surface, believing, wow, we can get good control. You can, but here's how, even in the laboratory, you know, not good spray coverage just proves the point of, well, the spores are applied, they land in areas that the copper wasn't, which allows them to germinate into the stomates, kind of grow within the leaf, and sporulate back out. Okay, so we understand it is labor intensive, it's time sensitive, but that is the best example where the coverage is key. Okay, so even not getting good coverage on a potted plant with the product leads to symptom development and spore production. Okay, so there's some other things that we wanted to look at as well. What other things could be maybe more of a friendly chemistry, more, you know, OMRI certified even, and these were products where it's anti-transpirant called VaporGuard. There was a product, Not Nice to Bugs, that have a variety of essential oils. And so what if we apply something like that and the potted plants get inoculated? What do we see? What do we find? Can they offer any protection? And this is just a quick summary wrap up with some pictures. You can see here that after the just symptom development, spots and spore production of a water control offering no protection on the leaf, comparing it to a standard fungicide all the way to the right, offering good control. Well, some of these products have some potential. It was good to see, I mean, we didn't really observe any kind of phyto effect, but it does show some potential. Now, this is just the early stages. More work is to be done, but at least it shows some promise that maybe some other things can be added to the arsenal for control of CLR. Okay, to get into field testing. So this is definitely where, boy, collaboration and the teams are invaluable as far as the amount of work with all of the application and the data taking and the et cetera, et cetera. So you all know who you are. We thank you so much. We say a FUFA for that. We have 2 years of data at a farm. This is actually, okay, so again, yes, I apologize, these next 4 slides. So this slide and the follow-up for 2022, I just wanted to give a kind of the overview, big picture of what we were doing, what we were collecting, and what the results were showing, okay? So here's the elevation of this farm up here. From the high point to the low point, we went in, actually did 3 reps called blocks, 1, 2, 3, labeled tons of trees. Here is the treatments. And last year was actually looking at different application methods, just the backpack versus the mist blower. Having all the labels and what it corresponds to. So things like having no treatment at all, having water and the surfactant, doing a control of badge, serenade, and double nickel as a rotation. Okay, looking at ProBlad, having this instill, so a different formulation of copper with serenade and double nickel. Cafidac, the Preaxor translaminar, and then a few of the experimental systemics that IR4 is studying, okay? This is the first application date of 2022, occurred the end of May. Then the second application was approximately 8 to 9 weeks later, and then continued to follow label recommendations. And for the ratings, essentially, the incidence rating for the entire tree was taken over the entire course of the season, okay? The rating scale, I'll point out, 0 being nothing. 1 is less than 5% incidence, 2, 5 to 10% incidence. I know it's a lot to digest, and even more so with this one slide. Really try to just look at the overall trends of this average tree incidence over time. And these are all 3 blocks combined. So this is all of the treatments combined to form one graph. So it's based on all of these colors, these various lines. The controls, again, up here, remind you what these controls were, including a no treatment control. Here, the rating scale, and starting here for 2022, week 13 was actually the end of August, going to week 23, the beginning of November, trends increasing in incidence, as we are getting towards harvest, which aligns with everything Melissa also sees in the field, and grouping into, well, what seems to work better, keeping incidence lower, these groupings as far as Preaxor and the true systemics, IR4 systemics. Here's some of the others with the rotations of double nickel, serenade, and then not so much looking up here with the ProBlad, Cafidac again, which is more, nutritional based products. But I wanted to point out, even though you do see increase and you can kind of see trends of separation based on active ingredients and the mode of action, here's this incidence line, 1, which is less than 5%. So for most of the season, most of whatever control measures were being applied kept them pretty well in check. Okay, even here with the scale, it's stretched out just to show the differences on the treatment. It's less than 10%. Okay, very promising. Really showed hope. Okay, going into 2023 now, the same kind of setup. We eliminated some of the treatments. We added a couple more. We increased the size and the treatment. So 3 blocks again, 3 reps, but spraying more trees, taking the ratings over time, looking at how much spot and spore production. And for 2023, it essentially started in June. And with this graph I'm going to show you going through October, still spraying, taking data, also harvest data, just to show you then the first application. And again, following the labels and recommendations. Okay. So 2023 data in a nutshell, same kind of thing. What we're trying to do here, this rainbow circle, the first product, the first spray for all products, okay? So took initial incidence rating of the trees on the 1st of June. You can see everything to start less than 5%. First sprayed occurred, and then these are the observation times. So if this is 0, 2, 4, 6 weeks to October, and what do the trends look like? And again, you know, I've marked here as far as the Preaxor working great, the systemics showing a lot of promise. But again, with this line, even in 2023, or you look at the end of the season, less than 10% incidence. So good control is possible with a variety of techniques, as long as really it's the coverage. We're going to stress that coverage is key. Okay, I'm wrapping up. I know that's probably like, oh, too much. One slide to show you, we are involved in variety testing. Here's the susceptibility, lots of spores being produced. We are looking at material. Well, is it highly tolerant? Is there true resistance? To be announced, and stay tuned for Dr. Tracy Matsumoto's talk probably in the new year. My favorite, natural enemies, really fungi, killing fungi. So we see this in the field. We're not happy. Those are just spores of Hevea. That's coffee leaf rust. When we see this, we are happy because this is a fungus growing on all of these spores. So mass collection. Thank you so much, everyone involved in this. Blaine, Lionel doing million isolations, getting a variety of interesting looking fungi to work with that are associated with CLR in the field. What are they doing? This is what we're on to now. We know what to call them, what their names are. What are they doing? There's a variety of antagonistic action, but this is what we're looking for. We want spores to be dead, which is really the goal for all management. Okay, combating CLR. This is the final, you know, this is the collective message and has been really from the beginning. It is a continuous epidemic. You have to manage it accordingly. There is no one silver bullet approach. It definitely is integrated pest management. There are short-term studies and long-term strategies. We know what race we have, race 24. We don't want to have new races evolve here or have new introduction of races. So it's very important to do all that we can do to manage. I'm just going to put this plug in, not as a scare tactic, but more so as a, do I see something? Definitely reach out. If you see fungal growth on green berries, please. These are things we do not have here. We do not want here, but at least to have a visual. If you see incredible stunting on trees or crazy patterns in the leaf, okay, if you see these very strange discolorations along the leaf veins, okay, or strange internal discoloration, please reach out. With that, I'll say thank you so much for your time. I will try to answer questions here, or please reach me. Here's my email. Thank you to our funding sources. You know, there's a lot of great just reference material out there, including the Best Beans app. And thank you so much for your time. Appreciate it.
Lisa, there are a couple of questions in Q&A session. If you could go to that tab and
Sure.
questions, that would be great. Thank you.
Okay. Well, the first question and the second question, thank you for your questions. Why no harvest for 45 days after application? Why only 2 applications allowed per season? Great questions. This really is just based on the manufacturer and the label. So I really have nothing to add to that. Sorry about that. You know, if that it's not a satisfying answer, we definitely can get back to you. But as far as residual, I'm guessing that's only an educated guess. The harvest, you know, post-harvest interval, probably. But beyond that, sorry, I can't contribute. So that's the Q&A. Okay, here's another question. Will thinning out the infected leaves help with the spread? So definitely good airflow, you know, cutting down on standing water, which is necessary for those spores to germinate, yes. Thinning out, that's where now you're really weighing, I guess, the pros and cons. You need enough good leaf material, right? Good photosynthesis, good growth, to have a good bean production. So I'm not sure if you're saying just good pruning, selective pruning, you know, not having rows overlap, definitely would be favorable to cutting down the problems with the disease. Okay. Where did Race 24 originate? That is from, well, genetically most similar to the cousins in Latin America, Caribbean, does exist in South America. It is the one we have. You know, when you talk about, I guess, the aggressiveness or so it's one thing to be a pathogen. So a pathogen can cause disease. Then there's various levels of what is termed virulence or aggressiveness. And Race 24 tends to be known as aggressive. There are other races that will affect far more varieties. So that's why it's key not to have new races evolve or the introduction of other races here. So thank you for that question on the Race 24. The question, thank you, Puhaku Coffee. The product had the 45-day wait time for harvesting. That's Preaxor. Okay. Are those systemic? And again, that is really categorized as the translaminar. It is not a true systemic. Hopefully that answers your questions. So, you know, definitely if you'd love to chime in or ask, and if not, I know that's that was a downloading of a lot of information to digest. Please, again, feel free to always reach out through emails best. Happy to provide more info or answer more questions, or you get off and you think, oh, what did that mean? Definitely reach out.
There's one more question.
Oh, okay. Okay. Hi, John. Any systemics approved for coffee not at the moment, not in Hawaii. For use on coffee. This is the critical work being done by the University of Hawaii Manoa IR4 program. Dr. Chen, Julie, they're definitely getting great results. You know, we help support that. So hopefully soon, sorry, I don't know the time frame, but I'd say hopefully soon there will be an option for a true systemic, which for the non-organic will be definitely good to have part of your toolbox.
That looks like you have answered all of the questions. Thank you so much for those who attended. Lisa, thank you so much for the presentation. It was super informal and informative. We will have the recording posted on our website if anybody needs to revisit this. Yeah, thank you so much and happy Thanksgiving.
Thank you. Bye-bye.